So far, we've been talking about passive transport, which means we're dealing with a situation where there is no need for an input of energy. When a molecule moves down its concentration gradient, this is a process that does not require an input of energy. It's spontaneous. But a lot of times, functioning of our cells requires that the cell forces molecules to move against their concentration gradient. The cell moves these ions from a place where they are less concentrated to a place where they are more concentrated. And this is a process that requires energy. And we call this active transport. Now, there are two types of active transport. One is primary active transport, where the source of energy is the molecule of ATP. Remember ATP, that the fuel of the cell. So an example that is shown in this image is the sodium-potassium pump that is present on the membrane of your cells, and it helps set a specific concentration of sodium and potassium ions across the plasma membrane. And basically, as a result of the work of this sodium-potassium channel, the concentration of sodium ions, which is represented by these orange figures, is higher outside the cell than inside the cell. You see, you have more of these orange thingies outside as compared to inside. And also, as a result of the work of this channel, the concentration of potassium ions is higher inside the cell as compared to outside the cell. So for each 3 sodium molecules that this channel, that is pump and channel, that pushes outside the cell, it brings in 2 potassium ions into the cell. So it creates a concentration gradient for sodium and potassium. And as a result, since you're pumping 3 sodiums out and 2 potassiums in, there's also going to be a buildup of negative charge inside and positive charge outside. So it kind of, this pump works to make our cells into a battery. Okay, so how does it work? When these sodium ions attach to the pump, the ATP comes in and binds to this protein. It's a transmembrane protein, and it's also a channel. It changes the shape of this protein, so then it spits out 3 sodium ions. Then on the other side, this channel also can bind potassium ions. It bonds 2 potassium ions, and when ATP breaks from being ATP to ADP and phosphate, it changes its conformation again. It changes shape, and then it allows the cell to bring in these 2 potassiums inside. So you can see that the cell is forming a gradient for both sodium and potassium. Potassium is more concentrated inside, sodium is more concentrated outside. And as you're sitting here, your cells are doing this, even though you probably are not doing much activity. This is happening in your cells all the time. So this channel is actively transporting sodium outside and potassium ions transporting them inside, and it requires the energy of ATP. The other type of active transport is called secondary active transport, which requires no ATP, and it actually, especially in this particular case, is taking the advantage of the fact that sodium is building up outside the cell. So you can think of this as a dam. We know that a water dam, you have this really thick concrete wall, and behind of it there is a buildup of water. Now, if you allow that water, if you put an opening inside the wall of the dam, what happens? The water will rush in really fast. And actually, this is how through dams work, the hydroelectric power. We have one in Las Vegas, close to Las Vegas. So you let the water rush in, and that rushing of water turns a turbine that turns into electricity. So a similar process is happening here. We are taking advantage of the fact that on one side of the cell, there's this buildup of these sodium ions, and there are places inside the membrane which allow this sodium ion to pass and go down their concentration gradient. And this is an energy releasing process, just like you release water from a dam. So then this molecule that binds sodium, that allows it to pass through the membrane, because remember, ions cannot just pass through the membrane by themselves. They need a channel or a transporter. Now, glucose will hitch a ride and takes advantage of this energy releasing process. So the cell can keep bringing glucose molecules from, let's say, the bloodstream. And even though there's plenty of glucose in here, the cell can keep bringing them in. So that's an example of a secondary active transport that doesn't require ATP. So the processes that so far we've talked about are for really smaller molecules, ions, and things like that. But what about when the cell wants to take a big bite? Well, this process is called endocytosis, and it involves the plasma membrane folding inwards and taking something and gulping it inside. It's just like how you eat, except you don't have to really fold your mouth. Your mouth has the opening in it, but the cell is covered with a plasma membrane, and when it's time to eat, it needs to fold in, and then this thing pinches, this folding pinches into a vacuole or a vesicle that is inside the cell. So endocytosis has different types. We have one that is phagocytosis, which involves taking really, really big bites. And this happens specifically with your immune cells called phagocytes, where they basically clean up. They're the first line of defense in your body. And when you get a bacteria coming inside your body, they just gobble up this bacteria and take them in. And the vacuole goes in, and guess what it's going to fuse with? Yes, it's going to fuse with a lysosome, which has digestive enzymes. Pinocytosis is when the cell takes small bites, so it takes in smaller molecules, not as big as bacteria, and again, it takes in through pinching the plasma membrane inward, and then it takes the vesicle where it's supposed to go. Receptor-mediated endocytosis is different from these two in that there is specificity. Just like when there is a lock and key, and you can only open the door with a key that is specific to a lock, this is similar. So when during receptor-mediated endocytosis, the cell takes in specialized, specific molecules and not just about anything. So there are specific molecules on the surface of the plasma membrane that is a match with the molecule that the cell wants to take in. See, it doesn't take in the blue ones, it only takes the star-shaped ones. And then the process of internalization is similar with the other two kinds. The cell pinches inward, but the difference is that places where these receptors are landing is covered in the inside of the cell with molecules called clathrin. So this is just basically, that's how the cell decides where it's going to put these receptors and then bring in those molecules that it wants. So this is endocytosis. Now here's a video of a cell actually taking up. This is a cell, these are bacterial cells, and it's a phagocyte, see? And it's taking in the bacteria inside. Then exocytosis is when the cell wants to get rid of something. So whenever the cell wants to get rid of things that it doesn't want, or when the cell secretes something outside into the outside of the cell. So this is, for example, the way nerves talk to each other or nerves talk to muscle. There are these neurotransmitters that come from nerve cells and go and signal either another cell or another nerve cell or a muscle cell. So that's called exocytosis, and you can think about this exo comes from exit. So again, a vesicle comes, it fuses with the plasma membrane and spits this thing out. If you remember, we talked about this when we covered the endomembrane system. So here's an animation. Here's a vesicle coming in. These phospholipids are going to find each other. And then basically the vesicle becomes one as the plasma membrane. And then you got these goodies that are going to be spit out into the extracellular fluid.